Welcome. I hope you find what I have to say interesting, which is about the transition away from animal use in biomedical research and testing. And I think it's important at the outset to remind ourselves that this has a few different aspects. There's animal ethics, of course. The use of animals in research and testing causes a great deal of suffering and often death, all for the potential benefit of humans. There's the question of science. Are we doing the best, most translational, most human relevant research and testing? And of course, this leads to human ethics, because if we're not doing the best, most translational, most human relevant research and testing, then we have to ask if we're causing harm to people by persisting with suboptimal or even downright irrelevant research testing and models. As well as by resisting or avoiding better, more translational methods of research and testing, we have to ask if we're approving harmful drugs and chemicals because they appear safe in animals. Are we losing potentially safe and effective drugs in humans because they harm animals in tests? And are we failing to understand human diseases sufficiently because animal models of those diseases bear little or no resemblance to them? Thankfully, we're now in an age where we hear regularly about the benefits of so-called NAMs, new approach methodologies. This is something we really used to dream about 15, 20 years ago. And these are, for example, advanced cell and tissue culture methods that are more physiologically relevant for actual human beings, that can reflect human biological variation, which are often patient-derived to better model human disease variability. And for example, these can be augmented by artificial intelligence and computational modeling, clinical research and more, providing the most human relevant preclinical research and testing platform that's ever been available to science. But today I'm going to focus on a reminder of why we need these methods, why we need this best ever platform, why these methods are superior to animal use scientifically, not just ethically, and why there is so much urgency to shift away from animal use and to accelerate the development of NAMs and their use in place of animals. In short, we must always be mindful that using animals has serious scientific flaws, which are pushing science to move forwards and away from animal use very quickly, and actually much quicker than is happening at the moment. So on to my talk. What I'm going to do is present an overview largely of my work over the last 20 or so years, but also some of the very important work of others, outlining some of the huge amount of evidence that's now available showing that animal research isn't good science, along with some examples of how science is moving on, how it's doing better both ethically and scientifically, how biomedical researchers are transitioning to human-focused research in many different areas. Now, over the next 40 minutes or so, there's going to be a lot of slides, a lot of information squeezed in, because they're taken from dozens of talks on specific papers, specific research projects that I've given over the years on lots of different topics. The reason I want to do this is because I really want to illustrate how much information is out there to show that animal experiments right across the board in a very wide range of areas are often of alarmingly poor human relevance, often to a degree that makes them counterproductive. And this results in harm to humans, as well as to the 150 million or so animals used in labs every year around the world. So Catherine mentioned a little bit about who I currently work for. I want to expand on that a little bit. I first joined the Physicians Committee when I was still in laboratory research way back in 2002. I did a project for the group on how drugs women are exposed to in pregnancy can be harmful to their unborn children, and how testing drugs for this was not well predicted in pregnant animals. And I spent 7 years working for the Physicians Committee as a consultant, along with one or two other groups. But I joined the Physicians Committee again just back in January this year as its Director of Medical Research. And it has many thousands of medically qualified supporters amongst its 1 million or so supporters internationally, working for preventive medicine, to promote better health through diet, but crucially, appealing for more focus on clinical research and better and more humane research methods in the laboratory. So this slide is actually just to show how the work I have done over the years has been published in peer-reviewed scientific journals as book chapters. I'm not simply talking about an unfounded opinion on science with little or no evidence, save the odd anecdotal example. So the work that forms the basis largely of what I'm about to present and that illustrates the basis of the argument for replacement is accessed, read, downloaded and cited. It's used, it's connecting with people, and I think that's a very important point to make. So on to some examples of this evidence now, which I'd like to remind you is a relatively superficial overview of each paper that it refers to, taken from dedicated talks, each of about 30 or 40 minutes. But I hope the key points will be of interest and will get through to you and will prompt some thoughts and discussion. And the first example comes from several publications on genetically modified or GM animals, driven by one major consideration. And this is that the rise we've seen in animal research over the past 20 to 30 years is solely due to the use of GM animals, genetically modified animals being used in place of non-genetically modified animals. And I think it illustrates a simple but alarming point. There's an assumption, and it's been argued, that this rise in the use of GM animals is due to them being better, being more human relevant of biological systems and human diseases. But does the evidence show that they're better? Well, I think largely the answer seems to be no. And these are just some of the areas of research in which many papers have been published showing that GM animals have failed to translate to human biology and or to reflect the human situation. And you can see that there's some big examples in here: neurodegenerative diseases like Alzheimer's and Parkinson's, function of the immune system, which is of course important in infectious disease research, diabetes, visual and problems, deafness, organ transplantation, and so on and so forth. There's some really, really big examples. And the reason for this degree of failure is actually simple and obvious, and I'll get onto this in a little while. Genetic regulation is complex, and we have more than enough evidence to know that if you duplicate, let's say, a genetic mutation in a different genetic and biological environment, it often doesn't and can't recreate what you see, for example, in a human individual with that mutation. Taking a genetic fault, putting it in a mouse and expecting that to inform you about what that gene does and what's gone wrong and how it's exerting its effects in humans just simply doesn't work. And there was a recent example of this in the magazine Vox, which has hundreds of millions of readers, just in October. And this was on attempts to model autism in mice for the purposes of research. And the article heard from people involved in autism advocacy and autism support, as well as from biomedical scientists, some of whom were researching autism and neurodevelopmental conditions, none of whom were complimentary about the relevance of this approach to the study of human autism. And I suggest, if you're interested in this, just Google it and read it. I really recommend it. It's an outstanding article that shows some of the problems that we're facing with applying animal research, even using genetically modified animals, to human biology and human conditions. Now, among all of the science and the curiosity about translation to humans and clinical benefit, I think we always have to remind ourselves of the ethics of the animals being used in their tens of millions and of what they go through as laboratory tools. And this is to show how many GM animals suffer and how much. At least half suffer some degree of pain, which is often serious, or death. And these are just some examples taken from a catalogue of GM mice that you can buy metaphorically off the shelf. And you can see some really disturbing things here: developmental abnormalities, hearing loss, sight loss, involuntary movements, paralysis, brain hemorrhages, and so on and so forth. The point is that many GM animals are bred to suffer from the moment they're born and until they die. And this is a serious ethical issue, of course. Now, moving on from GM animals to the use of animals in neuroscience research, if you like, how the brain works and what it can mean when things go wrong. Now, this is a huge ethical concern because so many neuroscientific experiments involve non-human primates or monkeys, and many of the experiments are highly invasive and involve significant suffering. And this is what led me to publish an in-depth review of this field back in 2016, which was published in the journal Atla, and more recently as a chapter in this book, Non-Human Primate Welfare. Now, this review concluded that, based on evidence in almost 200 referenced papers, there is a widespread overstatement of the contribution of monkey experiments to human neuroscience, with little or no substantiation of their claims. There is a widespread exaggeration generally of the human relevance of neuroscience research involving monkeys. There is a widespread failure to link knowledge that comes from basic curiosity-driven scientific research to actual benefits for humans, anything at least beyond speculation. And at the very same time, there is a playing down of the contribution of human research to neuroscience, the significance of what that human research has achieved, the powerful and ever-improving performance of non-invasive methods, the scope of what can be done with them, and with invasive research in humans. There's an underestimation of the significance of species differences between monkeys and humans and how these differences confound the application of monkey research to human neuroscience. And there's a reluctance or at times a refusal to acknowledge the genuine degree of suffering involved for the monkeys used in neuroscience experiments and the impact of the stress and the distress those monkeys endure on experimental results as well as their welfare. And what all of this does together, of course, is to skew the important harm-benefit analysis process that is meant to be central to any justification of animal research. And we argued that the harms are much greater than are accepted and the benefits for humans much lower than are claimed, even often non-existent. And in fact, a review commissioned by funders of research published back in 2011 agreed with much of what we said, even though it was conducted and influenced by those with an interest in monkey research. And they accepted, for example, that in neuroscience, in most cases, little direct evidence is available of actual medical benefit. In other cases, the panel was of the view it would have been possible to carry out the work not using monkeys, but with ethical human-based research. In vision research, an area that uses a lot of monkeys and it often involves very invasive research with high amounts of suffering, the actual and potential medical impacts of most of the studies was low. And what they concluded based on these sorts of findings was that further reviews of the outcomes, benefits and impacts of primate research should be carried out periodically, and that has been resisted. That has not been carried out. Now, our paper also made the case that powerful techniques are available to keep neuroscience research, as I've said, an area in which many monkeys are used and which often has severe consequences for them in a human perspective. Now, there's a lot of text on this slide. I don't expect you to read it all, but I wanted to show that many techniques and approaches such as scanning and imaging machines and other non-invasive ways of tracking brain activity are immensely productive and crucially not affected by species differences between humans and monkeys. And they're also used in many of the areas in which monkeys are heavily used, and also that they're providing increasingly detailed data at high temporal and spatial resolution, and all in a human context without the ethical problems of invasive research using monkeys and of trying to extrapolate data from monkeys to humans across species barriers. Now, notably, one of the claims made by monkey researchers is that you cannot get sufficient resolution using these techniques, so you have to use monkeys to study down to the level of single nerve cells. Well, firstly, there are many neuroscientists and neurosurgeons that disagree, who believe the methods available for human research are sufficiently powerful. And for example, I recently read a new paper reporting spatial resolution using MRI down to the sub-millimeter scale, which has been an aim for many years. This is increasingly hugely powerful to enable researchers to keep their research in a human perspective. And secondly, single neuron studies are done in humans. This is a book I have on that very topic. Many hundreds, if not thousands, of experiments have been done involving research volunteers undergoing brain surgery, for example, for epilepsy. The take-home point is that there are many, many examples of neuroscience research that used monkeys and for which it's claimed that avoiding using monkeys is impossible, but in which using humans and/or human tissue and cell cultures not only is possible, but is actually superior science. And the bonus is that, as I've said, we know we have to keep a human focus because it's unreliable to apply monkey data to humans. And there are many and increasing species differences in gene expression between humans and monkeys, which affect the function of neurons and the brain as a whole. And I'd like to give you a few examples of these. So for example, we know that even when humans and monkeys share genes, very, very often those genes are expressed differently. They do different jobs in different ways, and this really, really matters. And we see thousands of genes that we share expressed differently that affect brain structure and function. And we even know a little bit about the mechanisms behind these gene expression differences. So many of you will have heard of microRNA. These are little nucleic acid molecules that can turn genes on and off or switch their expression up and down. And it's known that as many as a third of hundreds of these molecules diverge significantly between humans and monkeys. And there are dozens of them present in one species, but not the other. And these affect the expression of hundreds and hundreds of genes sometimes. And you don't need to know what this is, but there's a process in gene expression called AI editing, and this occurs at a much higher rate in humans, particularly in the brain. And the point about it is that these differences affect the brain, they affect neuronal functions, and they affect the prevalence and the pathology of neurological diseases. So gene expression differences are extensive, and they really do matter. I'm going to move away from neuroscience now and move on to animal testing of new drugs, which, as we know, is ostensibly to protect human safety. Now, we looked at doing a project on this way back initially in 2009, and it was largely because there was no evidence, and I would argue there still is no substantial, robust evidence to show that testing new human drugs in animals works, is predictive, has a scientific basis. So we set out to do our own study using publicly available data, and we had thousands of examples of data that we could use in our analysis. And what we showed a number of different things in numerous papers, but among other things, we showed that when a monkey test suggests that a new human drug may be non-toxic, or if you like, safe, this adds almost no evidential weight to the existing probability based on other tests and data that the drug will be safe or free from toxic effects in humans. And we could quantify that. So if you have a proposed new human drug that, given existing data and other tests, we think is 70% probable to be non-toxic in humans. If you then do a monkey test with that drug and the monkey test suggests there's going to be no toxicity, how much does that raise that probability of 70% safety in humans? Well, we found it raised it just by 0.4%, from 70 to 70.4%. This is not significant, and it's a huge cost, both financially and to the monkeys used in the testing. Now, we also looked at this for dogs, which is the other common second species, in addition to a rat or a mouse used in drug testing, and found that a similar test in dogs raised the probability from 70% to just 72%. In other words, no or very, very little evidential weight is being added by these animal tests. Fortunately, there's now increasing pressure on many stakeholders on many sides to change this, to move away from animal testing, and actually a lot of goodwill to do that, and a lot of appreciation of the need to do that by many stakeholders. So it's a real sign of progress. Moving on again, as part of a campaign that I worked with for a while against using dogs in medical experiments and testing, I authored a review that included some very elegant research at Emory University in the US, where people's companion animals, people's pet dogs, were taken into the lab and trained to sit still for a little while in MRI machines while their brains were being imaged. And the neuroscientists involved have shown very advanced cognitive and emotional capacities in dogs. And these included things like this. The dogs don't just know what we're saying, they don't just know a lot of human words. They can distinguish between tones of voice and recognize different emotions in human speech. They can also interpret facial expressions. They can sense how somebody is feeling, what sort of mood they're in, what sort of intent they have by their facial expressions. And more importantly, they can link both of these things, tone of voice, patterns of speech, facial expressions to really work out how someone's feeling, what someone's intent is. And cognitively and neurologically, this is a very advanced thing to do. Neuroscience has shown that dogs experience positive emotions, that they feel empathy, that they can sense sadness and fear, that they demonstrate genuine and deep human bonding. Now, much of this should be obvious anyway to anyone who's ever owned a dog, who's ever really known a dog, although of course this knowledge hasn't stopped their use in invasive experiments that have caused an awful lot of suffering. But some neuroscientists have concluded, based on this type of experimental evidence, that dogs have the same level of understanding and awareness as young human children, and that should mean we rethink how we treat them, whether we should have dogs and be using them in laboratory research and testing at all. Now, I'd like to very quickly whiz through some other evidence, so hold your breath, but I'll be as quick as I can. I think this is just important, again, to convey the scale and the breadth of the evidence showing that we need to do something else. So we've been looking for an HIV/AIDS vaccine for decades, and we still don't have one. Now, I did this original analysis back in 2008 and showed at the time there'd been approaching almost 100 very diverse different vaccines that had shown promise, safety and efficacy in animals, often in monkeys, and until it was stopped in the US in chimpanzees. Now, the problem is these 85 different vaccines had been tested in nearly 200 clinical trials, and not one had worked in people. And I updated this back in 2016, and the number of clinical trials had increased greatly. But even now, heading into the year 2025, we remain without an HIV/AIDS vaccine that works in people, even though we have numerous candidates that have showed promise, safety and efficacy in non-human primates. Another issue which is still underappreciated and which not just impacts animal welfare, but also experimental data, is stress. And the point is that animals in labs are stressed unavoidably, chronically, and it stands to reason. It's an unnatural environment. The animals in these unnatural environments are going through often invasive experiments that cause stress and distress. And this is demonstrable. These are some of the physiological effects of stress experienced in labs by animals. And as you can see, it affects many physiological systems, many of them often the physiological systems being investigated in the experiments. And what it means is that any data often will not be applicable even to wild living individuals of the same species, never mind across the species barrier into humans. Now, some of these stressors, things that are causing this stress, can be mitigated to some degree, and there's some work, there is some good work being done, some efforts to try and make things better for animals in labs. But the point is that many of these stresses cannot be mitigated, and actually often attempts to enrich cages, provide foraging, toys to play with and so on, can actually be counterproductive and cause or exacerbate stress rather than make it any better. And this is perhaps my most read, most downloadable, most cited paper. It's really engaged people, and it's an important point to make. Now, I want to talk about something that's not done briefly, that's not done in the US anymore, which is chimpanzee research. I was involved in this campaign for a number of years, which was to try to stop federally funded chimpanzee research in the United States. And this was ultimately successful when the Institute of Medicine called an inquiry in 2011, at which I submitted and presented my work, as did others. And it helped lead the NIH to end federal funding of chimp research in the United States. And I published many papers outlining why this should end and how it would benefit not only the chimpanzees, but also human beings looking for more translational research to be done, and actually for the scientists doing that research. I'd like to run through a few examples, and I think the point is this: if chimpanzees, our closest genetic relative, pushing ethics to one side for the sake of argument, if chimpanzees can't be a good model for human beings, then it's very, very difficult to argue that any more distantly related species can be good models. But I'd like to just briefly run through the evidence. Very quickly, we saw that contrary to claims being made, often by chimp researchers, chimpanzees were almost never used in cancer research. The reason is because chimpanzees rarely get cancer. And even when they do, there are important differences compared to humans with cancer. In hepatitis C research, we saw, if you look at the graph on the left, that over a 30-year period, chimpanzee research had declined by about a third, while over the same period, the use of human-specific methods had increased very, very significantly indeed. That's the graph on the right. Many things that had been claimed could only be studied for hepatitis C in chimpanzees, it actually turned out with a little bit of effort could and were being studied using human-specific methods. And we also reported why chimpanzee research wasn't reliably informing human hepatitis C. As is the case with HIV/AIDS, while chimps could be infected with the virus, there were major differences in the pathology and the course of infection in the disease. This is humans on the left, chimpanzees on the right. In other words, something very different is going on when you infect chimpanzees with this virus than when you infect humans with it. And we also looked at why chimpanzees more generally and more widely were a failure in research. And it was claimed that they had to be a good model because they were almost identical genetically to humans. But actually, when you look more stringently, they're not as closely genetically related as they were claimed by chimpanzee researchers. And the same is the case for monkeys. They're closely related, but not as closely related as is claimed. Now, the point is, these figures might still seem impressive, 89 to 94% genetic identity, but these specific genetic differences are associated with many diseases and processes. In humans, for example, neurodegenerative diseases, Alzheimer's, Parkinson's, the function of the immune system, and infectious diseases like HIV, autoimmune diseases, cancers, and many others. Further, even when humans, chimpanzees, monkeys share genes, as I mentioned, those genes are often expressed differently with huge consequences. And this slide shows that in many organs and tissues, the brain as a whole, parts of the brain, liver, kidney, heart, and so on, a large percentage of genes that we share, a large number of genes, hundreds, sometimes thousands of them, are expressed differently with major consequences. So I'm going to start wrapping up now. I think that this evidence and much more evidence like it alone means we should really think twice about using animals as models in research for humans altogether. There is an urgency to move away from this. There are insurmountable species differences that really matter. But we have so-called alternatives, and the alternatives we have are amazing, and I'd like to quickly go through what some of these are. Now, many of these come under the term NAMs, New Approach Methodologies, and they include things like using human cells and tissues of many kinds which are grown in the lab. Now, human cells and tissues have been grown in the lab for a long time. I used to use them about 25 years ago, but they've developed and improved and become even more relevant to human tissues and cells and organs actually in a human body. And this is the type of improvement that you really can't do with animal research. So you can see we will have all, I'm sure, heard of organ-on-a-chip technologies, and this is one of those organ chips in the photograph on the right-hand side here. So many different, often three-dimensional cultures now with real physiological structures and function can be grown, for example, including on chips, tiny little chips with circulatory systems, with metabolic factors, and so on and so forth. And these can be linked together and known as body on a chip. And these sorts of technologies are already widely being used, particularly in research, but increasingly in drug discovery and development. And they're being constantly improved, and they can only get better. Things called organoids are being grown using human cells, often patient-derived, and these function like tiny little microscopic human organs, hearts, lungs, pancreas, and so on and so forth. Now, there's a really nice example of how this can help animals and humans, and this is a paper showing that human liver-on-a-chip technology can predict the harmful effects on the liver by new drugs much better than testing using animals. Now, in this paper, the authors report the retesting of 27 drugs for which human liver toxicity is known, either the presence or the absence of liver toxicity. And this retesting, which was blinded, so the researchers had no idea if the drug they were testing was toxic or not to the human liver, used human liver chips produced by a well-known company called Emulate, and they reported 87% sensitivity. In other words, the correct detection of almost 9 out of 10 drugs that were toxic to the human liver, despite those drugs having shown no toxicity to the liver in animal tests. And these are false results that resulted in almost 250 deaths and 10 liver transplants. And they also showed 100% specificity. In other words, no drugs were flagged as being toxic that actually were not toxic. And this would permit ongoing development after testing, reducing the likelihood of potentially safe and effective new human drugs being terminated unnecessarily and incorrectly. And on the basis of these results, it was proposed that using human liver chips in this way could increase R&D productivity by $3 billion per year for the pharmaceutical industry. And the wider use, along with other human organ chips, could similarly generate approximately $24 billion per year due to increased R&D productivity. Amazing networks also exist and are being set up and improved constantly to give researchers what they say they want, to facilitate the supply of human tissues to researchers, which can replace animal tests, as well as provide valuable research material for many diseases and biological processes. And the example on this slide is post-mortem brain tissues contributing to the understanding, for example, of multiple sclerosis and Parkinson's disease, and helping find new therapeutic targets for them. And we also shouldn't overlook the power of computers, which can model human organs and physiological systems, and in some cases have been shown to predict the human toxicity of industrial chemicals much better than animal tests. And artificial intelligence is also helping to prioritize existing drugs for repurposing or retesting existing drugs to see how they might be effective therapies for other diseases and illnesses. I'm sure you'll be relieved to hear that I'm almost there. I think this is the penultimate slide. I've given just a small number of examples, actually a relatively small number of examples of the evidence that is out there to generate evidence for a shift in how science is done. There's plenty more where that came from, but I can point you to that on the next and the last slide. But before going to questions, I want to address very quickly a question I'm often asked, which is gaining traction. It's the subject of sessions and panels at many conferences recently, and that is, what is stopping people from leaving animal research behind and moving on? What is impeding how the progression of this transition and giving scientists help to maintain a human focus in their research? And actually many reasons for this have been suggested, including by animal researchers who are keen to make the shift or who have made the shift. And reasons are diverse, but include simple habit, entrenched views, sometimes the hubris of animal researchers not wanting to be wrong, not wanting to leave the models that they have forged their careers behind, poor funding to help people shift away from animals and towards alternatives, bureaucracy, unfair validation requirements of alternatives, and so on and so forth. The good news is that these obstacles are being voiced and heard, discussed and appreciated, and crucially now addressed. In other words, things are being put into place to help researchers make their research better, more humane certainly, but more human relevant and translational also. And more people and organizations are getting on board. For example, the NIH is now accepting recommendations from its advisory councils to further the development of NAMs based on, quote, tremendous promise for helping us better understand fundamental biology to advance human health. Just last year, the director of the NIH launched a new program to accelerate the development, validation, and widespread use of these NAMs. And the FDA, the regulatory body that approves new drugs for human use, is working with NAMs providers and developers to incorporate them into their own workflows, things like human liver and intestinal organ chips to allow the better prediction of liver toxicity, as we've seen, and also human drug absorption than from animal tests. And just to finish off, also on another positive note, it's my traditional final slide, a collage of my furry and feathery friends, past and present, enjoying their lives, who remind me every day of the need to make science animal-free and human-focused. One, so that animals can enjoy what life is about, agency, freedom, and fun, but also two, so that people can reap the benefits of better, humane, human-relevant scientific research. So thank you so much for your attention. I really appreciate it, and I look forward to your questions and hope we can have a productive and interesting discussion. Thank you so much.
Thanks a lot, Jarrod. That was very interesting, as usual. There are a lot of questions, so I will start. So the first question for you is, how far are we away from really having enough non-animal methods to move away from animal experimentation? What's your
It's a
vision?
really good question, but unfortunately, it's a how long is a piece of string question. I think there's a real diversity of views on this. I'm probably at one end of the spectrum. I think, as I tried to allude to in my talk, I think the situation is so bad with animal research that we should have moved away from it much more quickly and started significant efforts to move away from it a long time ago. I think we're certainly in an amazing position now. It's far from perfect, but the crucial point, it's all relative, is much better. We're in a much better position than we are still trying to extrapolate animal data to human biology and human disease. So I think, yes, we've got some way to go, but we would get there more quickly if we embraced and used and developed and imparted confidence in these methods by concerted efforts that are starting to happen. So hopefully this is the tipping point and it will accelerate from here on in.
The next question is, what's your stance on chimeric generation of organs for human transplantation?
I think we have to change our mindset and be as human specific as we possibly can be. I think if we try hard enough, if we think hard enough, if we talk to enough people, and get enough expertise, there's always an optimal human focus that can get us out of, that can answer a particular problem and do the best science and the most translational science. So I think it's just about changing our mindset and actually forgetting about animals, starting with a clean slate, a clean piece of paper, working out what we need to know and how we can do things and how we can do it in a human perspective. There's always a way, and I think that mindset is starting to gain some traction.
Yeah, I think it's interesting to see how much focus is there still on xenotransplantation, where it would be way easier to educate and inform the general public about organ donation,
I think so. I think, put quite simply, if there's much effort and funding had been given to investigating various ways of dealing with this huge problem, and we all have sympathy for this problem, we'd be in a much better place. I think the money has gone to the wrong side of the argument, and it's one that's thrown up obstacle after obstacle, and when we're not hugely further forward after all of these years because of all of those obstacles, there are better options.
Yeah, I recall I went to a xenotransplantation workshop like almost 20 years ago, where they discussed the same problems as they have today. So it's interesting.
Exactly.
So the next question is your data about the little added value of non-human primates and dogs in toxicology published? The person is asking about the example with the 70% growing to 70.2% after non-human primate testing.
Yeah, it's actually on the last slide there. Maybe I've stopped sharing, but it's on my ResearchGate page, or email me. We actually published a number of papers and a number of letters in response to queries from the Association of the British Pharmaceutical Industry, to industry groups that had repeated our analysis and actually shared some of our conclusions. There's a lot published about it in great detail. So if you have a look on my ResearchGate page, or if you email me, I'll be very happy to send you. It was all published, I think, starting back in 2013 and in subsequent years. Yes, it's all out there.
Thank you for sharing your experience. I believe that economic implications of animal research are also very important. We are certain that NAMs target the main problem of using animal-free models, but do you think inclusivity could also be achieved through NAMs?
Yeah, that's a really important point. I think what's been really pleasing, I think just from my own subjective experience over the last 20 years, is that all of the aspects that we knew would be important have come together, and they're all working together. So the animal ethics, the human ethics, the better science, the better research, the funding, the increase in R&D productivity, the jobs, the economic benefits. everything's coming together now, and people are starting to work together and collaborate because they see common goals. And I think, where this used to be adversarial in many ways and people just fighting and arguing all the time, many people, many groups are now working together because they see those common goals, and this is why it's now starting to to really take off. And I think it doesn't really matter what those drivers are for the different the different stakeholders, the different subsections of the people involved. It's just that this really seems to benefit everybody and everything involved, and people are realizing that. And if it's money, it doesn't really matter. Money makes the world go round.
Yeah, maybe an interesting observation, I'm sure you share that, is that academia is a little bit lacking behind, but industry is totally on board with the new approach methods. Do you want to speak to that a little bit?
Yeah, I find this really interesting because I remember years ago there was a very friendly difference of opinion, just wondering who would go first, whether it would be academia leading industry or whether it would be industry leading academia. And it's been very interesting to watch that. I think there's now so much overlap between academia and industry that they're learning from each other, and I think that can only be a benefit. I think whoever's dragging whoever along, or helping, to be more positive, whoever's helping whomever up and do the right thing and do better science, it doesn't really matter. But what really does seem to be happening is that people's eyes are open now and they're seeing the possibilities, and they're jumping in, which is fabulous to see.
Yeah, there was one question earlier about on-site training, because obviously that's a big thing, or there's a lack of training opportunities at the moment in the new approach methods. And unfortunately, it doesn't look like academia is focusing on that, to restructure and redeploy funding is something really difficult. Do you have some insights there about where can you do training?
Yeah, I think having spoken to people and having thought about my own experience, having supervised PhD students and talking to them about this, there's still kind of what has always brought science on, which is learning from your peers and the enthusiasm of your peers who might have taken steps in these particular fields. So you'll get in at coffee time in the lab or journal clubs, or even just talking on campus or whatever. People will say, I was reading this paper, or I've got this kit and I'm trying it, or I'm stepping up my tissue culture and so on. And people just enthusing each other. That really seems to be starting to happen. But I think you're right. Money always talks, and I think there's now an increasing appreciation that you will do the best for your researchers in your country, in your state, or whatever, if you fund them, if you give them the help they need to follow their curiosity and to try things out. That's how scientific progress has always been made. And I think that's starting to happen. But the key thing is just the interest and the enthusiasm, and that spreads. And I think we're starting to see that have some effect in academia now.
Okay, you're very positive. That's great.
Yes.
There's one question that says, I keep hearing from the animal industry or those defending it that the technology needed to replace animals doesn't exist yet, or isn't capable of doing what we need it to do yet. Those who say this, what are they referring to? The cost, the technology, and how can we push back on those sorts of sentiments?
That's a really great comment and question. I like it a lot. I think, again, the benefit of talking to diverse scientists has been just breaking out of a particular way of thinking. If you're trying to replace a particular line of research or a particular experiment in an animal with something else, then you will run into difficulties. If you start with what you're ultimately trying to achieve, what you need to know to do what you want to do, ultimately higher up the chain, and then you can work out a way to do that. You can do that much more easily than trying to replace an animal experiment. So I think that idea of replacement is people are starting to change what that means for themselves and for science. It's not replacing an animal experiment with a different way of doing that experiment. It's looking at a higher level of what you need to know and how you can get that information. And there's always a better human-specific way to do that. And the reason is simple, and I quote this often, but it's something that I love, and it was a neuroscientist said, if you stick with using animals, there is always, no matter how human relevant your particular approach seems, there's some biological difference somewhere waiting to trip you up and bring it all down. And I think people are starting to realize that you know, if you start off with it with a human perspective from scratch, designing your research, you will design a much better research program and not even have to worry about replacing animals because you're doing better human relevant research.
The next question is about FDA and EMA. Why are they not, you know, accepting the new approach methods, or are they slowly accepting them? What is their requirements for fully substituting or replacing animals with NAMs?
Yeah, I think there's a lot of encouraging news. I think the frustration is always with the pace of change for many of us. It's, you know, we can see the change, attitudes are starting to change, the practice is starting to change, that people are becoming interested, they're kind of feeling their way here. Some of us think it's too slow, and the evidence demands that this should be happening much more quickly. But there's always been a thread throughout this, I think, for a long time, of the difficulty, the major difficulty in all of this is probably the regulators. And this is not an overt criticism of the regulators. It's, you know, their heads on the line. You know, they are going to be asked all of the questions if something doesn't work, if something ends up you know, falling down at some point. So I think people can understand that the regulators are having to be careful. That said, there's a lot of positivity there. They're starting to work with the NAMS developers to incorporate their technologies into their workflows. And I think, you know, they've taken those first steps. And once they see for themselves right in front of them that this is a really great step to take, and it's going to serve them as regulators really, really well as the industry and the patients that they serve, then again, there'll be a critical mass is hit, and that will take off too. So the steps are being taken. I think we have to understand to some degree that they're being careful about this. Yes, they could accelerate this, and I think they should. But the steps are being taken, and I think it will take off in the next few years.
So there's one question about cardiovascular research, and there's one researcher saying that she's been working with organs on chips for 10 years, but we are reproducing simplified models of human pathologies. They still needing a validation. And so she's asking, what are your suggestions in furthering, in this case, what cardiovascular research?
Yeah, obviously that's a very specific question. I think what I would say is what I've started to see over the last few years is researchers, perhaps even more than ever, learning from other researchers. I think I've already touched on this. And I think this is the point. We're seeing it in the you know, the Dutch effort, the helpathons, pulling researchers together to talk about this. Researchers who currently use animals, who are really intrigued by the new methods, who really have a desire to move to a more human focus, and just getting together and talking about what they're trying to do and learning from people who are using the technologies that can help them. You know, get together and work out ways to do it. So I think that's probably going to be really, really important in this transition phase, if you like, is researchers learning from each other and the experience of each other, especially ones who have some experience in these new methods with heart organoids and organ on a chip technologies and so on and so forth. So I think it can be, as you know, just as simple as finding the right people to talk to and putting a couple of hours aside to talk over a cup of coffee. I've seen that work in person beautifully, and it's very powerful.
And as you obviously mentioned in your talk, you know, what the animal models won't help us if they're not translatable. I think we always have to remember that Even,
Yeah.
even so, of course, we all agree we don't have all the methods yet, the human-based methods, but it's still the way forward compared to using models that might even be misleading.
I
Yeah,
think
so
that's a really good point. Just very quickly and simply, it's you use the best available model to you, and just because one model that you might be used to and might have used for a number of years is more complex, and you can maybe get more data from it, you always have to remember that ask the question: is this a good model or not? What does the evidence show you? And the evidence is now we're in a privileged position now. People like us, where we've been making this argument for years, and we have so much evidence to draw on now to point towards the poor human relevance, the poor translatability of these animal models. That there's always a good reason to just to use the most appropriate models, and it's always human.